Hello everyone, so today we'll be getting started a bit on the endocrine system, and we'll start today with hormones. So endocrine system is a system that's important to maintain a those sort of chemical messengers, unlike the nervous system that uses some sort of electric, no, I shouldn't call them electric. What sort of messengers? Neurotransmitters, yeah. Instead of using neurotransmitters and changes in charges, uh, endocrine system uses hormones, which are more chemically based. Many organs secrete at least one hormone. Primary endocrine organs secrete only hormones as their main or only function. And there are also secondary endocrine organs, which secrete hormones as long as well as additional functions. So hormone is a messenger that travels through blood to reach the target. Must be produced by one cell to find and activate protein receptors on another cell. And three, travel within the circulatory system. So you need to have all of these three rules in order for something to be a hormone. It has to be produced by one cell, binds to a protein receptor on another cell, and activate protein receptor on another cell, and you travel in the circulatory system. Now, the similarities between the endocrine system and the nervous system, let's say similarities, yes, differences. They both have chemical messengers. They have coordinating roles in homeostasis and feedback, and also in allostasis. And the differences are the modes of transport, transport speed, transport duration, and localization. Hormones can first be classified into three different classes. One of them, you can call them amino acid derivatives. These include epinephrine and norepinephrine. And what's key about these hormones is that, uh, sorry, give me a sec. What's key about these hormones is that norepinephrine and epinephrine, I believe they are lipid based. I'm gonna check my notes for a quick second here, actually. Amino acid hormones are an amine hormone, and they are secreted. They're stored in vesicles. The key thing is that they're stored in vesicles. And once the calcium levels rise, they then are released. And they are created through the adrenal gland. And let me check something. Adrenal gland, or specifically in the adrenal medulla in chromatin cells. And yeah, and the second class is about peptide hormones. They're synthesized from as larger inactivated proteins by ribosomes on the rough ER and are activated through cleavage as a pre-pro hormone. They are also, let's see where, they're also stored in larger vesicles and released when calcium levels rise, similar to, I should say, uh, what's the other type? When calcium levels rise. And yeah, they are very similar to type of amino acid hormones. released. The main thing is that they are synthesized differently. And the third type are lipid derivatives. This is modifications of cholesterol in the smooth, and they're made in the smooth endoplasmic reticulum and the mitochondria, and they're synthesized on demand. Transported in blood via carrier proteins. But the interesting thing about them being transported in blood is that because they're also lipid soluble, they are able to pass through directly. Another case, actually in class 1, is thyroid hormones. They're made by modifying amino acids on a large protein. In this case, they're made by adding iodine to thyroglobulin. And they are lipid soluble. And they diffuse once they are cleaved. And once they're cleaved from thyroid globulin, they diffuse in blood. Because they're lipid soluble, they have free diffusion and transport in blood on carrier proteins. Lipid soluble hormones such as thyroid hormones and lipid derivatives, these are generally able to just directly they are directly able to act through intracellular receptors and alter gene expression. So, cellular receptors, gene expression. While hydrophilic proteins, or we should say the amino acids and amino acid derived hormones and peptide hormones, they have to either enter through a GPCR, G protein couple receptors, or enzyme linked receptors to act onto their signal. Now, these are the ends, and these connect through essentially just secondary messages. They act and do secondary messengers and stuff like that. Whereas the lipid soluble ones alter cell activity directly. Many hormones participate in negative feedback loops where there's a sensor, integrator, and an effector, where maybe parts of them just don't like each other, and the produced product will affect how it is later on. For hormones can also participate in allostasis and preparing things in the future. And there's various interactions that might happen within hormones. I guess one thing you should note is orders. A first order feedback loop is when the sense organ is equal to the sensing integrator center. And there's a effector. So there's an order like this. Second order is when there's sense organ. Let me see. Sensing, integrating center, sense organ that acts upon an integrating sensor. And then we have an effector organ. This is the second. And the third would just be, well, no, let me see. Oh no, this would be a direct. This would be a first order, direct first order. And the second order would be, let's say, a sense organ as an integrating center, endocrine organ, and then effector. This would be a sense kick order where it goes through another organ. And the third order would be if there's a sense organ, then it would go to an integrating center, another organ, and then the target organ. So I guess you can just count the steps in between to understand the number, the degree of which it is, and what it does essentially. Hormones can also interact with each other, additive effects, where hormone 1 plus hormone 2 leads to increased activity when working together. You have antagonistic effects. Hormone 1 increases, whereas hormone 2 decreases. You can have integrated effects, where hormone 1 activates a complex that is needed for hormone 2 to activate something else. You also have permissive, where uh, one hormone, let's say hormone 1, is required for this, and hormone 2 can't do anything without it. Now, aside from just discussing about types of hormones and the general rules, one example in the body is a pituitary axis. So the pituitary gland is an endocrine organ from the hypothalamus. The hypothalamus is a collection of brain nuclei with different functions. One of them is maintaining circadian rhythm. The second one is alertness. Third one, behavioral drives. Fourth one is autonomic function. And pituitary glands are just some sort of glands that dangle off of the hypothalamus. And these hypothalamuses and these kind of ideas just have effects. So you can have the anterior pituitary and also the posterior pituitary. Now, the posterior pituitary hormones are secreted directly into bloodstream and include oxytocin and ADH, whereas the anterior pituitary has a whole bunch, and these are not directly, these are releasing hormones. And there's a nomic for it, but I'll tell you guys later. The most important idea is that the ones in the anterior are mainly tropic. Let me see, actually. I guess I'll describe it a bit later. Vasopressin, oxytocin, that's we call OT. These are both, the one thing we should note is these are both peptide hormones, which means they are pre-made as pre-pro hormones. And also the fact that they are hydrophilic, having some implications as well. Um, and yeah, these have much different functions. Vasopressin ADH leads to water conservation, whereas oxytocin is for uterine contraction and milk letdown. Mm, You should note that there are five hormones here: thyroid stimulating hormone, acetylcortical trophic hormone, gonadotrophins, which is follicular stimulating hormone and luteinizing hormone, growth hormone, and prolactin. Well, no, I think the hypothalamus releases of releasing hormones, and then the anterior pituitary releases these hormones. The hypothalamus uh, controls the release of corticotropin releasing hormone, thyrotropin releasing hormone, uh, growth, growth hormone releasing hormone, growth hormone inhibiting hormone, growth hormone inhibiting hormone, prolactin releasing hormone, and prolactin inhibiting release hormone, which leads to these effects here. Oh yeah, and also gonadotrophin releasing hormone, which leads to these two pairs. Uh, all the releasing hormones are secreted by the hypothalamus, whereas the anterior pituitary secretes the actual hormones. And they have different targets, I guess we should say. Gonadotrophs, corticotrophs, somatotrophs, lactotrophs, and thyrotrophs. Most of them are just releasing, but the two exceptions are actually growth hormone. So growth hormone from the hypothalamus can have, oh, it's my pen out of ink. So soon. Okay, my pen is actually out of ink. Give me a quick second. Uh, I didn't expect it to run out this fast. Hormone and prolactin are unique because growth hormone, well, I should say both of them, the hypothalamus will secrete growth hormone, inhibiting hormone, and also growth hormone, releasing hormone. And these affect some allotropes here to secrete growth hormone later. And then the same thing is pituitary inhibiting hormone, pituitary releasing hormone. And then this affects lactotropes, prolactin releasing hormone. Mm. So also some cases about some HP excesses here. So let's talk about the effects of each of these, at the very least, and just draw them out. There's three accesses that are pretty important. HPT accesses. One is for HPT, HPA, HPG. So first one is uh, thyroid releasing hormone, cortical releasing hormone, corticotrophin releasing hormone, gonadotrophin releasing hormone. And this is from releasing hormone, and this is from pituitary. So you're gonna have thyroid stimulating hormone, acetylcortical, ACTH, and then FSH, and I guess LH. And then the endocrine targets are going to be the thyroid, the adrenal cortex. FSH is going to test both testes and ovaries, while LH is going to also test testes, ovaries. For LH, it's gonna, well, I guess I should say hormone 2. This is gonna select thyroid hormones. This is gonna lead to glucocorticoids. Testes for FSH is gonna lead to inhibin, whereas testes for LH is gonna lead to testosterone. Testes for ovaries is gonna lead to progesterone and estrogens. and FSH for ovaries are going to be need to inhibin and estrogens. So you see you have estrogens in both for FSH and LH, which is unique, but FSH just usually does inhibin. And the interaction is that these hormones, they're gonna inhibit everything here, also going to inhibit everything here, right? Negative feedback. And negative feedback generally occurs. Same thing with the case of growth hormone, releasing hormone, and growth hormone, inhibiting hormone. It's going to lead to more growth hormone, which is going to help growth hormone inhibiting, and it's going to inhibit growth hormone releasing hormone. And the product, when it works in the liver, somatostatins, it's also going to stimulate this, but it's also going to inhibit this. Now, we should talk about the idea of tropic and trophic. Tropic means regulatory. Imagine it's a tropic island and you're in control of all your emotions. That's what tropic is. Trophic means simulating division. So I guess you can assume that, I guess you can kind of imagine the tropic hormones are going to be in the anterior pituitary, whereas the trophic are going to be in the posterior pituitary. However, some anterior pituitary hormones are also trophic. The examples we can talk about, acetylcortical, ACTH, TSH, FSH, and LH. So these are some exceptions. And yeah, I don't think there's anything much else that should be discussed. Um, okay, let's talk about control of growth and metabolism. That's a big word. So metabolism, all chemical and physical changes in body tissue. And this relates to three things: mobilization and delivery of nutrients, basal metabolism rate, and anabolism and tissue growth. Basal metabolism rate is using and expending energy at rest. Metabolism is mobilizing nutrients and distributing it. And this just refers to growth. So let's talk about it. So mobilizing and direct regulation of BMR, this place is only done using thyroid hormones. Mobilizing, there's thyroid hormones as well, but there's also stuff like glucagon, um, insulin, cortisol, growth hormone. Whereas for anabolism, there's also growth hormone, there's also sex steroids, calcitriol, and all that jazz. So thyroid hormone affects BMR through affecting mitochondrial function because they are, remember, hydrophobic. They can act directly, diffuse through the plasma membrane into intracellular receptors, and they can also affect metabolic processes by changing gene expression. This they do in the nucleus. Many non-thyroid hormones also can affect metabolism. So in terms of mobilizing resources, let's list some of them. In the liver, you have epinephrine and glucagon, which both use fatty acids and glucose and mobilize them and turn them into energy. Oh, I should say stored fatty acids and glucose. So this would be glycogen or fat or whatnot. And then have, you can also utilize it directly using glucocorticoids and growth hormone and mobilize the same fatty acids and glucose storage. However, promote lipid use and stimulate gluconeogenesis. This means that they're going to make more new glucose and they're also going to promote the use of fats. This is what we call a glucose sparing effect. And this way, they're able to save glucose to use for the brain. That's really nice because the brain cannot use fatty acids, I should say. It cannot use fatty acids or ATP. So these things can store, um, they can synthesize glucose, fatty acids, and ketones. Growth hormone also has a function promoting growth in skeleton and bones and muscle. Uh, and they do this by increasing cellular proliferation, uptake of amino acids in blood. And this can be also happen indirectly using somatomedins by the liver, which when released can also have similar effects. Now, let me see what else we should discuss. Another thing is that these metabolic and non-metabolic hormones cooperate at different life stages. You have embryo and infancy, which uses more thyroid hormones. You have childhood, which uses parathyroid hormones and calcitriol. And you have puberty, which is mainly relying on reproductive hormones. Mm. And then you have two cases of hyperthyroidism and hypothyroidism. Uh, Both cases of hypothyroidism and hyperthyroidism, well, I say they should have different symptoms. Hyperthyroidism leads to generally being feeling more hot, increased active thyroid activity. So you should see increased metabolism. Whereas hypothyroidism, you're going to see the opposite. You're going to see lower metabolic functions and whatnot. And yeah, all that jazz. Thyroid hormones also have what we call pleiotropic effects because they have receptors on many different organs. You can see them affecting many different things depending on what they are. There's also a lot of endocrine diseases that can occur due to any cases regarding uh, underproduction or overproduction of hormones or sensitivity sensitivity regarding uh, sensitivity regarding hormone receptor. Sorry. Best example we have is diabetes. For diabetes, the key thing about diabetes is that pancreatic islet cells secrete two hormones. So we have the alpha cells and beta cells. Alpha cells secrete glucagon, uh, whereas beta cells secrete insulin, both amino acid hormones, by the way. So this should tell you that they are sort of vesicles, that, yeah, sort of vesicles where they're secreted, and also that they are created by modifying amino acids groups and such. And they have negative feedback loops to maintain uh, a normal glucose levels. Uh, and ATP, so what I should say is direct feedback is regulated by blood glucose. So, for example, if you eat a meal, this increases blood glucose, and yeah, directly affects the pancreas, which secretes hormones, and the glucose. So the glucose is basically going to receptors in the pancreas. Blood glucose in the blood will affect uh, pancreas. It will affect will affect beta cells. They'll affect the pancreas beta cells, and the blood glucose will lead to increased insulin secretion. This is with K. This is because glucose in blood is converted to ATP. Then the ATP is used to open K channels, which leads to depolarization. And then calcium enters, which leads to vesicle releasing insulin. And now insulin allows cells to take up glucose, and insulin helps reduce blood glucose in the liver by enhancing storage through glycogen synthase. You make more glycogen, whereas glucagon does the opposite. Glucagon releases glycogen in the liver into the bloodstream as glucose. And there's also, let's talk about type 1 diabetes in this case. Going to be a lot of wasting, meaning urination. Type 1 is destruction of beta cells in pancreas. These are the ones that make insulin. Type 2 is insulin resistance. Symptoms are high glucose in blood and urine for both. That's number one, but for the first one, destruction of beta cells can lead to drowsiness and lethargy. Type 1 can be controlled using injections of insulin. Type 2 can have multiple strategies. You can do injections, but you can also have lifestyle changes, uh, reducing dietary glucose, and whatnot. Let's go. Now we can get to the stress response. Stress is another important hormone axis. You don't have stress, well, you wouldn't be watching these videos. I wouldn't be here. You wouldn't be here. And yeah, it begins in the brain that categorizes the situation as stressful, and then it basically just goes into any neuroendocrine systems. Now, the two hormonal pathways for stress include adrenal medulla or the adrenal cortex. The cortex increases cortisol, whereas the medulla increases epinephrine. First, we have to talk about the general adaptation syndrome. So first, the resistance, let's say we have a graph of resistance. Stress resistance lowers a bit and then increases and then lowers again. So for the first part, is the alarm phase. And the second part is the resistance phase and the exhaustion phase. So let's talk about it. In the alarm phase, you have a fight or flight reaction. And the dominant hormone is going to be epinephrine, made by adrenal medulla. In the resistance phase, you're going to have recovery. The dominant hormone is cortisol, made by the adrenal cortex. The exhaustion phase is where prolonged and severe stress, where there really isn't a hormone and you're just kind of depleted of everything. The alarm phase usually goes to sympathetic activation. And I think you should probably review what sympathetic activation means, but remember it's a flight or fight response. During the alarm phase, you have both norepinephrine and epinephrine, leads to alertness and physical activity. Now, another case is cortisol. Cortisol and glucocorticoids, I guess you can say glucocorticoids, they lead to gluconeogenesis. They basically try to maintain the phase of resistance. They diminish immune function and fat catabolism in adipose tissue. So basically, they get rid of fats, increase glucose in liver. Remember the glucose sparing effect. And they also reduce immune function. And this happens in the adrenal cortex, specifically the zona fasciculata. And then, yeah, Rexabiles. It also inhibits thyroid stimulating hormone, growth hormone, and gonadotrophins, so G and RH. And then when you have constant stress, you have when a moderate stress is sustained over a long periods of time, you are going to get chronic stress response. And this is going to lead to changes in alarm phase response, which is affected with many negative conditions. One condition is actually chronic stress affects the hippocampus, meaning memory. So if you're ever stressing or you're ever studying while you're stressed, you should keep in mind maybe it's good to take a break sometimes. So how does this work? So it can lead to shrinkage and reduced activity of hippocampal neurons. So in the normal HPA axis, the hippocampus is a negative feedback loop where hippocampus releases, or hypothalamus releases cortical releasing hormone to the pituitary. This turns into aceto-ACTH, and then this stimulates adrenal glands, well, adrenal cortex to produce cortisol. And then when cortisol is increased, this signals the pituitary gland and the hypothalamus to inhibit this. But in a chronic stage, this is normal. But in a chronic stage, we're going to have less cortical releasing hormone, which leads to similar amounts of ACTH and a lot of cortisol. Let me check actually. It basically just has no negative feedback anymore. Increased cortisol releasing hormone. This is going to have increased ACTH, increased cortisol, and the negative feedback is going to be a bit gone. Managed. Oh, so yeah, sorry, I was blanking out a bit. That's because hippocampus inhibits CRH release, um, and amygdala activity increases CRH release, amygdala. So let's say it's high CRH, high acetylcholine, high this goes into this, then this inhibits the hippocampus and also inhibits the amygdala. So chronic uh, cortical, chronic stress, chronic symptoms of stress are gonna affect the hippocampus by inhibiting cortical releasing hormone, leading to increased hippocampus activity. Sorry, I mean it's gonna inhibit the amygdala. It's gonna lead to increased hippocampus activity because hippocampus leads to hypothalamus, inhibits CRH release. It's chronic, yeah. So yeah, let me check again actually. Well, I guess we can talk about it in a bit, but actually give me a second real quick. I need to find something that might help explain this a bit better. Okay, I found it actually. Let's see. Okay, let's talk about sex determination and development. That's one key thing about hormones, as we all know. First thing is Sex is what you have. Gender is who you are socially, trying to identify. Sexuality, who you love. So, in terms of sex, you have male, female, gender, male, woman, non-binary. There's many differences. You have morphological, chromosomal, whatnot. Chromosomal defined by XY or XX. And one gene on the Y chromosome, SRY gene, on the Y, responsible for male sexual development. And this pushes gonads to develop into testes, ovaries. Uh, at early developments, embryos all have two sets of gonads and internal ducts. And SRY, if you have you're SRY positive, this releases testes determining factor or SOX9 plus, and this leads to testes. And if you don't have SRY, then no, that's the determining factor. You're going to have ovaries. Support cells in testes, which are Sertoli, they secrete AMH, which develops Wolffian ducts. And this, what's it called? Wolffian ducts, whereas the Mullerian ducts degrade. And then if you have no SRO, no AMH, the Wolffian ducts degrade, and the Mullerian ducts turns into the uterus. I should say the Wolffian ducts uh, forms male internal genitalia, which basically is the vas deferens in this case. And another case is the secretion of steroid hormones on XX and XY. And XX, you have ovaries, you have testes. Leydig cells secrete testosterone, whereas follicular cells secrete estrogen and progesterone. Granulosa cells, and you also have germ cells. And the equivalent is Sertoli cells and also germ cells. So Leydig in particular are the same. Granulosa and Sertoli are also the same. Uh, morphological. The production of testosterone leads to androgens, which is testes, whereas estrogen leads to ovaries. And this leads to morphological sex due to androgen receptor signaling or not. And sex is a non-binary product. Another thing interesting is that sex testes formation leads to secretion of AMH and androgens, which masculinize internal and external respiratory system. And yeah, key things are that estrogen receptors and uh, estrogen receptors and androgen receptor expression are all in the hypothalamus and the amygdala, and also have effects on fetal brain development and are important in critical periods early on that can change how you think and you feel. During puberty, hormones increase into maturation. Hypothalamus increases GnRH, FSH, and LH. Both of them lead to gamete initiation, sex hormones release, and this leads to secondary sexual effects such as sexual drive, body hair patterns, red blood cell production, and whatnot. And that's basically the gist. Let's study this backside of this paper. Okay, now how about let's get into sperm production and delivery. Male reproductive system is composed of internal and external genitalia, and the function is to manufacture gametes, deliver them to ovum. Now remember about the testes being developed. We have many features. The testes also have a scrotum, skin and muscle. They have membranes and fascia. And they also have seminiferous tubules. Sperm are produced here. Sperm are formed within the germ cells of seminiferous tubules. So what you would look like. You were cut open a single seminiferous tubule. It's kind of like a tube again. You would first have the spermatogoniums, which are the most early developing cells, and then spermatocytes, spermatids, and then sperm, where they grow kind of their things here. You have nurse cells, which are much more different. And you also have some endocrine cells surrounding it. You have Leydig cells outside and Sertoli cells inside, which are nurse cells, which kind of nurse them. Yeah, so it goes from the order spermatogonium to spermatocyte to spermatid and sperm. This is due to one round of mitosis. So you would have two primary spermatogonium going to spermatocytes. You would have two spermatocytes. And then two spermatocytes, you have around a meiosis here. Meiosis one, you have two, what you call now four secondary spermatocytes. And then each of the secondary spermatocytes turn to spermatids. Uh, spermatogonium, spermatocytes, spermatids. So you would have, we have two spermatocytes here, and then you had four. And then each of them would be eight spermatids, and then you would have sperm. So in general, you would go from one spermatogonium to two spermatocytes, and then the spermatocytes will just divide again to form four secondary spermatocytes, and then the four secondary produces eight spermatids. And this takes about 2 months. You should note that this mitosis first takes 16 days, the middle part takes 24 days, and then converting to spermatids takes hours, really quick. But then the sperm maturing is 24 days as well. And this leads to development of, I should say, moving sperm towards a lumen of the seminiferous tubules. So they progress from spermatogonium, They move from the basal layer of the seminiferous tubules to the lumen, which, remember the diagram, they go towards here, maintaining contact with Sertoli cells. There's also, should draw out the areas of the sperm. You have the acrosome, which has enzymes needed to penetrate an ovum. Head, which contains genetic material. The neck, which doesn't really do anything. The middle piece has mitochondria and ATP for energy. And then the flagellum, the tail, that helps move them. And an anatomically mature sperm isn't necessarily mature. I'll show you the nomic 7 up. Seminiferous tubules, ependidium, vas deferens, ejaculatory duct, nothing, urethra, and penis. So first, they're produced in the seminiferous tubules. Ependidium, uh, in the epididym is when they have physiological maturation. Physiological maturation. They give the sperm immobilin, which leads them immotile, which helps them move. And they also have compact genetic material, maturation of acrosomal proteins, alter sperm plasma membrane. And then they're transported to the vas deferens for storage. And in the vas deferens, they're mixed with the they have something called capacitance, where they become capable of fertilization. This is mixed with seminal fluids. This happens in the ejaculatory duct. So I just think you highlight seminal fluids for motility. And there's a bunch of other fluids you should know. Seminal fluids activate sperm for motility. Prostate gland protects urethra from infection. So these fluids, bulbonus glands, and then lubricates penis and protects sperm from urethra. And then the penis conducts it to the outside of the body. And lastly, the penile tissue is actually innervated by both A and S and S and S parts. So as for the A and S, you have parasympathetic fibers, which innervates tissue, and they release NO, which relaxes smooth muscle, leading to an erection. And then sympathetic fibers innervates ependidymis, vas deferens, seminal vesicles. Sorry, so first they release NO, nitric oxide. Nitric oxide relaxes smooth muscle in penis, causing an erection. And the sympathetic fibers innervates ependidymis, vas deferens, and seminal vesicles to help move sperm and secretions. Lastly, you have SNS, which are the lumbosacral somatic motor neurons. This is the skeletal muscles at base of penis. Helps ejaculatory reflex. Okay, now. Another key thing, HPG axis controls spermatogenesis, and HPG axis, I should say, yeah. Remember this one? Gonadotrophin releasing hormone leads to LH and FSH. Then in the testes, this leads to stimulating and sertraline, testosterone, inhibin. Inhibin inhibits, has negative feedback onto FSH, whereas testosterone has negative feedbacks on Leydig cells and on gonadotrophin releasing hormone. Oh, no, 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 not Leydig cells. Only gonadotrophin releasing hormone. Leydig cells activation directs androgen synthesis, aka testosterone, and Sertoli cells also promote spermatogenesis. So they cooperate in their roles. So testosterone leads to sexual drive, bone and muscle growth, and also maintaining secondary characteristics, male sexual characteristics, maintain tissues and accessory glands of reproductive system. Sertoli also secretes ABP, androgen binding protein, binds androgens to the in seminiferous tubules. Express androgens. Androgens work through Sertoli cells to maintain spermatogonia numbers, promote meiosis, and adhesion of spermatids and whatnot. So, let's see. What's the next one we should go over? Oh, I should clarify. This affects both LH and FSH. Inhibits gonadotrophs in the, I should say, gonadotrophs in anterior-posterior. Okay, let's go over the ovarian cycle. Sorry, I'm gonna have to check actually. Okay, yeah, let's go over the ovarian cycle. So the ovarian cycle is a bit different. Well, I guess it's not that much different. It's kind of sharing the same steps, but contains of internal and external genitalia. They are designed to manufacture gametes and help the embryo. They develop similarly to the bipotential gonad. Mm, Archer ovary has a cortex and a medulla. And oocytes form from germ cells within uh, follicles. So you probably see an oocyte here, oocyte, and then you have a granulosa cell, then you have thecal cells. And oogenesis is a bit different because you have the oogonium, uh, make it turn into a oocyte, and then a secondary oocyte, and then ovum. But it involves only a single oocyte that's created at the end. Or wait, I should say a secondary ovum, oocyte, I mean, in the end. So oogonium, oocyte, secondary oocyte, and then this is the last one because during the division, well, I should say, the oogonium has mitosis here. The first polar body of one of the oocytes is actually thrown away. So the only one that matures is just one of them. This is completed in 5 months in utero. So all the oogonium, the number of eggs a woman has is already predetermined. Meiosis 1 is done monthly, every menstrual cycle around monthly, so it's around 14 days. And then meiosis 2, or just the process, or no, I should say the process of converting it to primary oocyte, or the process of meiosis 2 is going to take, well, the preparation here turning to the ovum, it's going to only take hours. And this has in parallel with the development of the follicle surrounding an individual oocyte. So what this means is primordial oocytes are just surrounded by follicular cells. Primary and primary follicles, or primary oocyte. And then we have granulosa, and then thecal. And then secondary oocyte, you're going to have a zona pellucida that you're going to see. And then you're going to have same in tertiary, you're going to have the corona radiata. Corona radiata, or it's being ejected. And at ovulation, the follicle ruptures. At ovulation, the follicle ruptures, releasing secondary oocyte and then transferring back to the corpus luteum. If it doesn't get fertilized. So, uh, hmm. Sorry, my throat is kind of scratchy. So, follicles that do not complete the ovulation cycle becomes atreic during the luteal phase, and this can lead to decreased fertility. Now, the HPG axis controls oogenesis and ovarian secretions, and the HPG hormone secretions vary across the ovarian cycle. Let's talk about it. GnRH, remember, goes into gonadotropes that secretes FSH, and LH leads to ovarian hormones. This leads to negative feedback loops here and here. So follicular stimulating hormones stimulates follicular proliferation. It helps secretion of aromatase by granula cells and also inhibin, whereas luteinizing hormone stimulates meiosis, ovulation, corpus luteum formation, stimulates secretion of testosterone by thecal cells and progesterone by luteum. First thing is inhibin provides negative feedback, as per usual. Estrogens also have a primary feedback role. They can either be positive or negative, depending on how much they have. If you have low levels of estrogen, you have a high negative feedback, where you have high levels, you have positive feedback. And the last thing, progesterone. Progesterone has negative feedback and is a primary role of negative feedback at GnRH. So when you have a lot of progesterone, you have less GnRH. Does this make sense? Okay. So oral contraceptive pills are gonna likely, or well, what they do is they're gonna want to increase the progesterone. And by increasing progesterone, well, you're not gonna have any GnRH, you're not gonna have any estrogen or anything, and you're not gonna initiate a sort of, you're not gonna initiate any sort of ovulation. So ovulation, remember, it releases a secondary oocyte into the peritoneal cavity. And the released oocytes are moved towards the uterus, which then is just a place I can shrink and move. But Before we get onto that, I think we should, I guess we should maybe, uh, no, I think this is relevant. So the oocytes move into the fallopian tube, and then are moved into the uterus. And then the uterus has a myometrium, perimetrium, protective layer, smooth myometrium, smooth muscle, and endometrium, secretory membrane. Endometrium is simple columnar and has a functional layer and a basal layer. What's important is a functional layer sheds during the menstrual cycle. During the follicular phase, it first follicular phase first sheds, then repairs, and then the luteal phase is about secretion by uterine glands. So the ovarian and uterine cycle are kind of in sync. So let's talk about gonadotrophin levels, FSH and LH. So what it looks like if you were to draw the graph out, follicular phase, luteal phase. Initially, you would have a GnRH pulse, GnRH pulse here that increases as you lead to ovulation. And GnRH leads to GnRH. First, you'll have a little pulse here, and then FSH dips, and then increases here, and just never really comes back. But then the LH goes extremely high like this. So why might this be so? Well, well, with this, let's understand that if you were to match this, ovulation happens here. And let's match it with ovarian hormone levels. So remember, FSH leads to inhibin, which decreases some stuff. So when you see the FSH pulse, you imagine, okay, inhibin is going to decrease the secretion of everything. Everything is going to decrease. But at the same time, what does LH do? LH secretes a lot of estrogen, and estrogen is going to keep on increasing. But at the same time, what secretes progesterone? Well, progesterone is also, well, let's see. Well, estrogen is secreted by the corpus luteum. Well, I mean, progesterone is secreted by the corpus luteum. So once the corpus luteum is promoted, so what happens is we're first going to see none of it. And then progesterone is secreted by the corpus luteum, which inhibits basically everything. But what's important is, so we're going to see increases in both estrogen and inhibin up until nowish, because FSH and LH are going to be at its all-time high, then they're going to decrease, right? But then they're going to increase again for the luteum formation and then decrease once more. So why is there this other increase here? Let's wonder. Uh, because the corpus luteum also secretes, let's see here. Once you form the corpus luteum, well, LH helps secrete. LH helps secrete. Let's actually label this. Well, estrogen increases because follicular cell proliferation, despite that there is negative activity from FSH, from inhibin, that or negative feedback from LH. there still is able, there still is able to manage. Mm, and estrogen and inhibin. Let's see why they are high. Oh, I see. After ovulation, estrogen rises due to production by granulosa cells within the follicle, even despite the decrease, the active cells in the corpus luteum maintain it. And inhibin is also increasing because of the FSH and LH that is secreted then. And this also matches up to the menstrual cycle. First shedding, and then healing, and then secretory phase. You need FSH first for follicular development, and then at ovulation you need high LH. So FSH first acts on for follicle development, and then LH has a spike for all maintaining ovulation. And then, yeah, increasing estrogen levels, negative feedback, which inhibits GnRH and LH release. And then after ovulation, there's there's an LH surge, mitosis, and whatnot. Ovulation, corpus luteum, the return of the leg, and then luteal phase. The ruptures turns back to ruptured follicle, turns back to corpus luteum, secretes. Oh, the corpus luteum secretes progesterone and estrogen, and exerts negative feedback that way. It was all done by the corpus luteum. Lastly, we need to know some muscles and what's involved in an orgasm. So, autonomic and somatic nervous system. Uh, so, autonomic nervous system, you have the engorgement of erectile tissue, and you also have lubricating secretions from vaginal glands. And so, sympathetic nervous system is, well, somatic nervous system is activation of pelvic floor muscles. Autonomic nervous system is smooth muscle in uterus and vagina. And those are just the cases that you might see it. For an orgasm, yeah, somatic nervous system is pelvic floor, smooth muscle is. Oh, yeah. You should talk about, okay, there should be a separation. There's arousal and also for an orgasm. So for an orgasm, there's smooth muscle and pelvic floor muscles, smooth muscle in vagina, uterus, pelvic floor muscles, or somatic nervous system. For arousal, there's engorgement of erectile tissue and also lubricating secretions from vaginal glands. And both parasympathetic and sympathetic uh, arousal should occur. Yeah. And I guess we should talk about, I guess we can say pregnancy. We'll say pregnancy and fertilization tomorrow or next time. But it's been a good chat, and I hope you guys learned something today. Have a nice day.